This is a basic introduction to immunology. My microphone is broken, that is why it sounds distant. What is the function of immunology? It is to protect the system against foreign microbes. However, sometimes the system goes wrong. What are examples of things that can go wrong? Autoimmune diseases, hypersensitivities, and immunodeficiencies. What are three examples of roles of the immune system? Defense against infections. The immune system recognizes and responds to tissue grafts and newly introduced proteins. Defense against tumors. What are the implications against defense against infections? Deficient immunity results in increased susceptibility to infections, exemplified by AIDS. Vaccination boosts immune defenses and protects against infections. What are the implications when considering that the immune system recognizes and responds to tissue grafts and newly introduced proteins? Immune responses are barriers to transplantation and gene therapy. What is the implication when considering defense against tumors? It is a potential for immunotherapy of cancer. How can you basically define innate immunity? Innate immunity is less specific. It is an early response. What are the principal components? The principal components could be epithelia, phagocytic cells, for example, neutrophils, macrophages, and natural killer cells, NK cells, and cytokines and blood proteins. What is one of the problems that may arise? One of the problems here with innate immunity is that it may not distinguish fine differences between foreign substances. What are examples of some of the players that may come into contact with foreign microbes? Some of the players would be epithelial barriers, phagocytes, complement, and NK cells. Let's think about some of the anatomical barriers. What barriers might you have in the eye? Examples would be blinking, tears, and lysozyme. What barriers would you have in the respiratory tract? Barriers here would include coughing and sneezing, mucus, ciliary action, phagocytes, and lysozyme. What anatomical barriers would you have in the GI tract? Examples would be stomach acidity, normal flora, peristalsis, antimicrobial compounds. What anatomical barriers would you have in the urogenital tract? Examples would be lavaging action of urine, acidity of urine, lysozyme, vaginal lactic acid, normal flora. What would be examples of the anatomical barriers of the skin? Examples of barriers for the skin would be the structural barrier between sweat sebum, lactic acid, propionic acid, lysozyme, and normal flora. What are the innate mechanisms within skin? The innate mechanisms with skin would be antimicrobial peptides, fatty acids, and sebum. What are the innate mechanisms within the mouth and upper alimentary canal? That would be enzymes, antimicrobial peptides, and sweeping of surface by directional flow of fluid towards stomach. What innate mechanisms would you find within the stomach? Innate mechanisms would include low pH, digestive enzymes, antimicrobial peptides, fluid flow towards intestines. What is a problem that may result from a patient with GERD who is taking proton pump inhibitors? One problem is that the natural innate mechanism would be destroyed because of the raising of pH. What innate mechanisms would you find in the small intestine? Digestive enzymes, antimicrobial peptides, fluid flow to the large intestine. What innate mechanisms would you find in the large intestine? Normal intestinal bacteria compete with invading microbes, fluid, feces expelled from rectum. What innate mechanisms would you find in the airway and lungs? Cilia sweep mucus outward, coughing, sneezing, expel mucus, macrophages, and alveoli of lungs. Let's talk about adaptive immunity. What are some of the main components of adaptive immunity? The main components would be B lymphocytes and T lymphocytes. What are lymphocytes stimulated by? Lymphocytes are stimulated by antigens, which are specific pieces of foreign substance. How would you broadly define adaptive immunity? Adaptive immunity is broadly defined as being a later response. It is slower and it's more specific, and it responds vigorously to repeated exposure to the same microbes. Generally speaking, what would be the result of adaptive immunity concerning B lymphocytes? In this case, adaptive immunity for B lymphocytes would result in antibodies. What would be the result of adaptive immunity for T lymphocytes? The result of adaptive immunity for T lymphocytes would be effector T cells. What are the two types of adaptive immunity? The two types of adaptive immunity are humoral and cell-mediated. What molecules in the blood mediate humoral immunity? This is mediated by antibodies that are produced by B lymphocytes. What do antibodies do? Antibodies specifically recognize microbial antigens, neutralize the infectivity of the microbe, and target microbes for elimination. What is the general function of humoral immunity? The general function of humoral immunity is to block infections and eliminate extracellular microbes. What type of lymphocyte mediates cell-mediated immunity? Cell-mediated immunity is mediated by T lymphocytes. What are two main examples of cell-mediated immunity? One example is phagocytosed microbes inside a macrophage. Another is intracellular microbes, for example, viruses replicating within an infected cell. Concerning a phagocytosed microbe within a macrophage, what would be the next step? The next step would be a responding lymphocyte, a helper T lymphocyte, which would then result in an effector mechanism, and the function here would be to activate macrophages to kill phagocytosed microbes. Again, I'll ask the question, what would be the function, the final function of phagocytosed microbes within a macrophage? The resulting function here would be to activate macrophages to kill phagocytosed microbes. Concerning an intracellular microbe, for example a virus replicating within an infected cell, what would be the next step? The next step would be responding lymphocyte, in this case cytotoxic T lymphocyte. What would be the result of the responding lymphocyte, in this case a cytotoxic T lymphocyte? The result would be the killing of the infected cell and elimination of the reservoir of infection. Now let's summarize humoral immunity and cell-mediated immunity. Let's think about the microbe involved, the responding lymphocytes, the effector mechanism, and the function. First, let's talk about humoral immunity. What would be the microbe involved? The microbe involved here would be an extracellular microbe. What would be the lymphocyte responding in the case of humoral immunity? The responding lymphocyte in humoral immunity would be the B lymphocyte. After the B lymphocyte responds in humoral immunity, what would be the effector mechanism? The effector mechanism would be secreted antibody. What would be the function? The function here finally would be to block infections and eliminate extracellular microbes. In cell-mediated immunity, let's consider two cases. First, phagocytosed microbes within a macrophage, and also intracellular microbes, for example viruses replicating within an infected cell. What would be the responding lymphocyte concerning a phagocytosed microbe within a macrophage? The responding lymphocyte would be a helper T lymphocyte. What would be the effector mechanism? The effector mechanism would be to activate macrophages to kill the phagocytosed microbes. Now let's think about intracellular microbes, for example viruses replicating within an infected cell. What would be the responding lymphocyte? The responding lymphocyte would be a cytotoxic T lymphocyte. What would be the function? The function would be to kill the infected cells and eliminate reservoirs